Hey everyone, this is chapter 36 of Campbell Bio, Resource Acquisition and Transport in Vascular Plants. So, an overview of resource acquisition is that water and minerals in the soil are absorbed by the roots, and then they travel up to the shoots of the plant, which are like the leaves, in this case, through the xylem sap. And then the water is lost through transpiration, which is a driving force for the cohesion tension hypothesis, which involves transpiration pulling up the xylem sap from the roots to the shoots. And then in the leaves and green stems, gas exchange occurs through stomata. During photosynthesis, CO2 is taken up, and then O2 is produced. The cells use some of this O2 for cellular respiration to produce ATP, and the rest is released into the air. Sugars are produced by photosynthesis in the leaves. So the photosynthesis equation is 6CO2 plus 6H2O plus light energy produces glucose, and oxygen. And then continuing on, the sugar produced by photosynthesis can flow in both directions. So it could go from the shoot to the root, or from the root to the shoot. It moves from sites of sugar production, which are usually leaves, or storage, which are usually roots, to sites of sugar use or storage. And also root cells carry out cellular respiration, exchanging gases with the air spaces of the soil, also taking in O2 and discharging CO2. Now, the different types of phyllotaxy are alternate arrangements, opposite arrangements, and whorled arrangements. Most angiosperms have an alternate phyllotaxy with 137.5 degree rotations between each leaf. And now the leaf area index is basically it's a ratio of the total area of the top surfaces of the leaves to the area of ground covered by the plant, which basically demonstrates how much area the leaf is able to cover. In pockets of soil with low amounts of nutrients, or with a high number of roots from the same plant roots will go straight ahead instead of branching. In areas with high amounts of nutrients, plant roots will branch out. So, short distance transport routes. The apoplast is everything that's external to the plasma membrane, including cell walls, intercellular spaces, and dead walls, which includes the xylem. So basically, in the apoplastic transport route, the materials just go through the cell walls and other dead cells. The symplast is the continuum of cytosol connected by plasmadesmata, which is the alive tissue within the plant. The transmembrane route involves taking transport routes that go through both the cytosol and the cell wall, so through both the symplast and the apoplast. There are various methods of solute transport across plant cell membranes. In figure A, which is this one, the plasma membranes of plant cells use ATP-dependent proton pumps to pump H+ out of the cell. These pumps contribute to the membrane potential and the establishment of a pH gradient across the membrane. These two forms of potential energy can drive the transport of solutes. So here, protons are actively transported out of the cell through proton pumps, which can establish a pH gradient, which can lead to more transport and diffusion across the cell membrane. In figure B, there is H+ and a co-transport of neutral solutes. Neutral solutes, such as sugars, can be loaded into plant cells by co-transport with H+ ions. H+ or sucrose co-transporters, for example, play a key role in loading sugar into the phloem prior to sugar transport through the plant. This will be elaborated more on later in this video. So here, the H+ and sugar are co-transported across the membrane at the same time, which could have implications for further reactions. Figure C, H plus and other ions are co-transported. Co-transport mechanisms involving H plus also participate in regulating ion fluxes into and out of cells. For example, H plus or nitrate co-transporters in the plasma membranes of root cells are important for the uptake of nitrate by plant roots. So here, H plus and nitrate are co-transported at the same time. In figure D, there are ion channels which simply allow ions to pass through the cell membrane where they regularly would have difficulty passing without the ion channels. And also the potassium ion shown in this figure is important for stomatal closing. In this figure, we see two examples of water potential. So there is an initial flaccid cell with a solute potential of negative 0.7 megapascals, as seen here. And then it's placed into a solution with 0.4 molarity sucrose. This gives it total water potential of negative 0.9 megapascals. And then the cell loses water to match osmotic equilibrium with its surroundings. As seen in this description, the protoplast loses water and the cell plasmolyzes. After plasmolysis is complete, the water potentials of the cell and its surrounding are the same. So this is an example of an osmoconformer. In this next example, we see an initial flaccid cell, which is at negative 0.7 megapascals. It is placed into an environment of pure water, which is 0 megapascals. So as a result, the water diffuses through the cell membrane, which ensures that there's osmotic equilibrium. As a result, the cell swells. Net uptake of water by osmosis makes the cell turgid. When this tendency for water to enter is offset by the back pressure of the elastic wall, water potentials are equal for the cell and its surroundings. This is also an example of an osmoconformer. To begin with, the endodermis is the innermost layer of the cortex in land plants. It's a cylinder of compact living cells. So here it's shown how there are various routes through which materials can pass through the cell walls to reach their final destinations within the plants. There is the apoplastic route, which is an uptake of soil solution by the hydrophilic walls of root hairs to provide access to the apoplast. Water and minerals can then diffuse into the cortex along this matrix of cells and extracellular spaces. And then there is a symplastic route. Minerals and water that cross the plasma membrane of root hairs can enter the symplast. So to bring back an aforementioned definition, the apoplast is the collection of dead tissues, right, such as the xylem or the cell walls. And then a symplastic root is alive, in contrast. The transmembrane route involves components of both the apoplastic and the symplastic route. It simply goes through both the cytosol and also various cell walls. As the soil solution moves along the apoplast, some water and minerals are transported into the protoplasts of cells and of the epidermis and cortex, and then move inward via the symplast. And then finally, the endodermis controls entry to the vascular cylinder. It basically acts as a passage that prevents materials from going in through the apoplast and makes them go through the symplast to ensure that toxins get filtered out, because the symplast involves selectively permeable layers. And then there's also transport in the xylem. Endodermal cells and also living cells within the vascular cylinder discharge water and minerals into their walls, which is the apoplast. The xylem vessels then transport the water and minerals by bulk flow upward into the chute system. So this also we call a definition, which is the cohesion tension hypothesis caused by transpiration, which was the loss of water through leaves. Root pressure is a force that helps to drive fluids upward from the soil into the water-conducting xylem vessels. Root pressure can be caused by the uptake of mineral ions into the root xylem, which leads more water to enter the roots, which in turn pushes water from the root to the shoot in an upwards direction. The cohesion tension hypothesis is the most widely accepted model for movement of water in vascular plants. It combines the process of capillary action with transpiration or the evaporation of water from the plant stomata. Basically, the first step, water is lost through transpiration. So, at first, the water lost is replaced by evaporation from the water film that coats mesophyll cells. But the evaporation of this film causes the air-water interface to retreat further into the cell wall to become more curved. This curvature increases the surface tension and the rate of transpiration. So then more water is lost. But in order to compensate for the water loss, water from the xylem is pulled into the surrounding cells and air spaces to replace the water that was just lost. So through this, water can be pulled from the root to the shoot. And building on this, the water flows from less negative pressure to a more negative pressure. The soil has a pressure of negative 0.3 megapascals, but the outside air, which is closest to the leaves at the top, has an air pressure of negative 100 megapascals. This is all due to the water loss incurred by transpiration at the leaves, and various other factors such as cohesion and adhesion in the xylem, and also the water uptake from the soil. Potassium ions have a significant role in stomatal opening and closing. The transport of potassium ions across the plasma membrane and vacuolar membrane causes the turgor changes of guard cells. So basically, when more potassium enters the guard cells, more water enters in turn to try to neutralize the concentrations. In turn, the guard cells open, which allow air to enter the plant. But then when the potassium ions leave, the water exits the guard cells, which leads to stomatal closing. Sucrose manufactured in mesophyll cells can travel via the symplast to sieve tube elements. In some species, sucrose can exit the symplast near sieve tubes and then travel through the apoplast, which is indicated by the red arrow. It is then actively accumulated from the apoplast by sieve tube elements and their companion cells. And then here, a chemiosmotic mechanism shows how the active transport of sucrose into companion cells and sieve tube elements functions at the molecular level. So H+ is actively transported out of the cell to an area of a high proton concentration. And then the proton and the sucrose molecule use a cotransporter to enter back into the cell. In this case, they would be passively transporting back into the cell because the proton would go from an area of high concentration to low concentration. This couples sucrose transport to the diffusion of H+ back into the cell. Positive pressure also contributes to how sucrose moves within the plant. So first, sugar is unloaded into the sieve tube at the source, which reduces water potential inside the sieve tube elements. This causes more water to enter in order to try to neutralize the concentration. And this uptake of water generates a positive pressure that forces the sap to flow along the tube. This pressure is relieved by the unloading of sugar and the subsequent loss of water at the sink. In leaf-to-root translocation, xylem recycles water from sink to source.